Hi, I'm Dr. Mike Zaretsky, Medical Director of the Colorado Fetal Care Center, and I'm here with...
I'm Dr. Sean McGrath. I'm manager of the Stem Cell Biobank and Disease Modeling Core in the Gates Center for Regenerative Medicine at the University of Colorado Anschutz Medical Campus.
And today we're going to be talking about gene editing technology called CRISPR. So Sean, tell us about CRISPR.
In its essence, CRISPR-Cas9 is just a molecular tool that scientists have adapted to modify DNA. It was first identified in bacteria as a clustered, regularly interspaced, short palindromic repeat, which is long and hard to say, so we call it CRISPR-Cas9. It is utilized naturally in bacteria to fight off viral infection by cutting that viral genetic material. Once it gets into the bacteria, it's actually an adaptive immune response. We've been able to take that and harness the ability of CRISPR-Cas9 to cut DNA and repurpose it to be able to modify DNA in eukaryotic cells.
So tell us a little bit more about how CRISPR works.
Yeah, as we use it today, CRISPR-Cas9 is really a two-piece system. The first piece is the Cas9 enzyme, and that is a protein that has the ability to bind DNA and then also cut it. The other piece of the system is a short RNA sequence. We call it the guide RNA because it binds to the Cas9 protein and then also has a seed sequence that is perfectly complementary to a DNA sequence of interest, and it can guide or home that Cas9 enzyme to that piece of DNA and cut it.
Nucleases and genome editing has been around for decades. What makes CRISPR unique?
Yeah, that's exactly right. One of the first nucleases discovered was zinc finger nucleases. That was in the 90s. Tau effector nucleases, or talins, those were found about 2009 or so. Both of them are DNA nucleases, so they can cut DNA just like CRISPR-Cas9. And in fact, they have been developed and are in clinical trials for various diseases. CRISPR-Cas9 is the newcomer on the block. Was discovered in 2010s or so in kind of the form that we use it today. And the real advantages of CRISPR-Cas9 over the zinc finger and talon systems is just how easy it is to use, how effective and cheap it is to employ.
So what is CRISPR used for?
So CRISPR, again, is basically just a DNA nuclease. There's other nucleases. They all have the function of cutting DNA. And that's really how they're used. If you want to modify DNA, you first have to cut it, and then you can employ the cell's natural ability to repair that damage to make changes to the DNA. So examples of what you could use CRISPR for, if you cut DNA within a gene of interest, you can knock out that gene. You can also utilize the cell's ability to do homology-directed repair. If you cut around a mutation, for example, you can correct that mutation and turn it back into a wild-type cell. In research, we're also using it in many different ways. You can fuse that Cas9 protein to various functional domains that we have been used to investigate many different things in the lab. So for example, you can fuse Cas9 to activators or repressors to turn genes on and off. You can fuse it to fluorescent domains and actually count loci within the cell. You can do DNA pull downs and start looking at how genes are regulated, for example.
Are there any safety concerns regarding CRISPR?
Yeah, so that's a really important question and a hotly studied topic in the field. There are some safety concerns. So Cas9 is a very specific technology, is very good at finding a particular piece of DNA and cutting that region, but it's not perfect. So the seed sequence in the guide RNA is 20 base pairs long, which is long enough that it homes it really to a particular region. But the genome of humans is very, very large, and so often there's another sequence that are different by only a base pair or a few base pairs. And that is sufficiently similar that Cas9 can recognize these off-target sites with only a few base pair differences and still cut it. Okay, so this is a concern because if you are trying to, let's say, correct a particular mutation, it doesn't do you any good to go somewhere else and create a new mutation that may cause further problems. So there's a few ways to that scientists and researchers are trying to fix this. We're learning more and more about how the system works. We're getting better tools to design guide RNAs, for example, with better specificity. We're also finding ways to modify the guide RNA to make it more specific. Also, people are trying the strategy of directly modifying the Cas9 protein. You can introduce mutations that make it more specific and try and prevent that off-target modifications that you don't want.
Is CRISPR being used to treat disease?
Yeah, so that obviously is the holy grail. That's what everybody is working towards. At the moment, there are no FDA-approved drugs or therapies using CRISPR-Cas9. The biggest impact that this technology has had so far is in research. Literally thousands, over 10,000 papers have been published since Cas9 was first characterized in the bacterial system in 2013. So the progress has been incredible. And there are several clinical trials that are actually ongoing and approved right now. So the first in the U.S. clinical trial was to modify T cells to try and target cancers in the body. Other examples of clinical trials using CRISPR-Cas9 is for inherited retinal diseases or also blood disorders such as beta thalassemia and sickle cell disease. But again, these are just in clinical trials, and there's a lot of ongoing efforts to try and develop further therapeutics. Now, you're doing some research with CRISPR-Cas9 as well. Can you tell us a little bit about that?
Sure. This is a pretty large collaboration of researchers, including yourself, who are experts in the field. My interest has to do with fetal therapy. Is there a potential role to correct mutations before delivery in order to ameliorate or improve outcomes for kids with disease? And the one that we're looking at is cystic fibrosis. And one single point mutation causes 70% of disease for kids with cystic fibrosis. So our goal is to see first whether we can target that mutation in utero to hopefully improve the condition in kids before delivery. Of course, this is only an animal model at this time, but the hope is way down in the future that perhaps we can get to this disease early on, even before birth.
So why is genetic correction of cystic fibrosis potentially a better approach than some of the current therapeutics?
Yeah, we're getting to the core of the issue. We're trying to actually correct the genetic mutation that leads to all the medical problems that these children and adults face. So the hope is that when we correct this mutation, that we reverse or we prevent, in our case, in our research, when we do this in utero, that we actually prevent the disease from happening. So the hope is that we can prolong lifespan beyond what we're currently at, which is, I believe, in the 40s these days, due to the advances in medicine that they're able to deliver to these children and adults through their lifespan.
So is in utero gene correction of cystic fibrosis something that could be available soon?
Yeah, I think we're many, many years away from this. However, we are beginning to do the work in the animal model, and we have collaborators, part of our research group, that have developed a cystic fibrosis sheep model, and also a model that will have the most common mutation for cystic fibrosis available to be working on in the lab. And the hope is way down the road that we can translate this into human use, but we got, again, long way to go. And that's the great hope, not just for cystic fibrosis, but for many other genetic mutations out there that have significant effect on our population. Thank you for listening today, and if you want further information, please visit us at the Colorado Fetal Care Center online, or
You can find us online looking for the Gates Center for Regenerative Medicine.